Since the discovery of penicillin by Alexander Fleming in 1928, humans have developed antibiotics that can effectively fight a variety of bacteria. However, there is still no good antiviral drug that can treat something as simple as a common cold. Most antivirals today work by targeting the virus itself. For example, Tamiflu, the best drug against the flu once infection has already occurred, works by binding onto a protein on the virus surface and preventing the virus from spreading to other cells. The problem with this is that it isn't hard for viruses to develop resistance against this drug. In 2007, a new strain of Tamiflu-resistant flu viruses emerged, rendering the drug useless. To solve this, scientists have taken a different approach to fighting viruses. Instead of attacking the virus itself, the drug alters its own cells to make them unsuitable for viruses to invade. Scientists have taken 3 ways to achieve this. The first strategy is to prevent the virus from maturing. A researcher from San Francisco called Vishwanath Lingpada discovered that some types of proteins can prevent a virus from assembling. Using this discovery, he and his sister started a company called Prozetta, and their research have shown that their approach can stop rabies, Ebola, influenza, and a number of other viruses. The second strategy, developed by Eleanor Fish from the University of Toronto, seeks to boost the immune system to help it fight against viruses. When we become infected by a virus, our body produces a type of protein called interferons, which help kill viruses in several different ways. However, viruses often replicate fast enough to stay ahead of the interferons, and others create proteins that can shut down interferons. So Fish and other scientists have developed artificial interferons that do a better job than natural ones. Synthetic interferons have shown to be able to cure some strains of hepatitis C and help patients recover from other viruses. If this is successful, synthetic interferons can become a drug that can be used to treat almost any virus. The third strategy, developed by Todd Ryder, is probably the most radical out of all of them. He is developing an artificial protein called Draco that detects viruses and then causes the infected cell to commit suicide. Viruses that develop resistance to antivirals would have no way of evading this protein. Many scientists are still skeptical about the feasibility of creating a wonder drug against viruses that could work as well as penicillin. Furthermore, antivirals may wipe out beneficial viruses in the human body, many of which we still don't know about.